day to all of you. This video is about the outline of the prokaryotic replication. So in this video, we are going to deal some terms and their outline overall view of the prokaryotic replication. And in next video, we are just going to see their mechanism. So in prokaryotes have a single origin, but in the eukaryote have several origin in the DNA through which the replication start. So prokaryotic replication in this region of the DNA, the ORI C contains 245 base pair long and have a two different conserved repeat sequence. So from the ORI site, the replication bubble forms by the unwinding of the DNA sequence or the DNA strand. So bidirectional replication and the unidirectional replication are the two methods by which the DNA will replicate and start the replication process. So in this figure, there is a two way of strand, lagging strand and the leading strand. Leading strand is a continuous, while the lagging strand have the short stretches of the strand, and which is away from the replication fork, and leading strand are the towards the replication fork. And these short stretches of the lagging strand called as the Okazaki fragments. So unidirectional of the replication fork. So difference between the prokaryotic and the eukaryotic fragments are the Okazaki fragments are large, where the Okazaki fragments are short in the case of the eukaryotes, and some other differences are here. So the enzymes and the factor involved with the prokaryotic replication are the primer, which is very important for the replication process, which start the replications, and the primase are the enzymes. So DNA polymerase can only make DNA in the 5' to 3' direction, and it is always in antiparallel. Second factors are the DNA polymerase, which is subcategorized into DNA polymerase 1, 2, 3. So 3 DNA and 4 and 5. So DNA 3 polymerase is very important for the replication process. So DNA polymerase 3 is discovered by the Thomas Kornberg, son of the Arthur Kornberg and the Malcolm Gafter in 1970. It has subunits like alpha, epsilon, theta, and 2 beta subunits, which have different function. So structure of the DNA polymerase 3 is like this. So proteins and the enzymes involved in the DNA replication. This chart is very important for the DNA replication. So we have to memorize it. Like topoisomerase in prokaryote, it is topoisomerase 1 and the 2, which are responsible for the winding and the unwinding process. And in eukaryote, it is called as the DNA gyrase. Helicase are the DNA B, and in this eukaryotes, MCM, that is mini chromosomal maintenance. CD6 and CD21 cell division cycle and polymerase alpha are the primase enzyme. So difference between the eukaryotes and the prokaryotic enzymes. So we can understand much better through this figure. Like these are the two strands of the DNA which are in the Y-shaped structure, which shows the unwinding region of the DNA, and there is several enzymes which involved and helps in the DNA replication process. So all about this, all outline view of the prokaryotic replication. Keep watching. Thank you.